Hello, I'm Abdul. This is one of the UTI series fourth video, and this is about the pathogenesis. So, what happened that there may be the bacteria. Usually, they are fecal flora, and it colonizes the perineal region, so it may go through the urethra into the bladder, and that time it will cause cystitis. But it may ascend via ureters to the pelvis of the kidney and collected system, and then it can induce an immunological inflammatory response and resulting in pyelonephritis and ultimately scarring. We in the text, if you read that there are certain mechanisms that which inhibit this intrarenal reflux of urine, but there are some in the upper and lower pole of the kidney, some papillae, which allow this, and this lead to the formation of the and inducing the inflammatory response leading to pyelonephritis. Hematogenous spread may be one of the causes implicated in a UTI, and it usually happens in endocarditis and bacteremia in infancy. Certain bacterial properties and certain are important in the pathogenesis, and if I will go a touch to it from the textbook, and actually it depends that the bacteria have this pili or fimbri in it. So there are two types. The type 1 is found in most cases of the strain, and they are blocked by the D mannose, and they're called mannose sensitive. And if they are not blocked by the mannose, the D mannose, we call it mannose resistant, and they are called type 2 fimbri. So E. coli, some strain express this type 2, and the receptor for this type 2 fimbri is a glycosphingolipid, and it is present both on the uroepithelial cell and red blood cell 1. So the gal1 to 4 gal oligosaccharide fraction is actually the specific receptor. I think there is no need to remember this, but because these fimbri can agglutinate by P blood group erythrocyte, they are known as P fimbri. So bacteria with P fimbri are more likely to cause pyelonephritis. So if you it's important in pathogens, if you say that those who have those bacteria which have P fimbri, they can cause pyelonephritis. And 76 to 94 of the pyelonephritogenic strains of have P fimbri, and that compared to the 19 to 23% of the cystitis strains. So how the cystitis strain different from the pyelonephritogenic strains? Because of the P fimbri. And I wanted to talk about these two names that you should remember, and because they are involved in the recurrent UTI, so I wanted to talk about it. This is the intracellular bacterial communities, and there is another term that's called quiescent intracellular reservoirs. So how it happens, and I will just give a touch to it, and that is that normally when an E. coli UTI, what happened that an E. coli bacteria comes and it binds to the mannose receptor on the umbrella cells. What these cells are, the cell that line the bladder. So the bladder lining cell take the E. coli into the cell, and there the bacteria replicate, and they form a bacterial community which we call intracellular bacterial community. We call it IBCs. So what happened that in IBCs, some of the E. coli changes into filamentous form, and because they don't divide, they fail to divide. How the bladder get rid of these IBCs? Number one is that usually the bladder shed the lining cells with the IBCs, and this is one way to get rid of the IBCs. But what happens that some of the IBC are released from the cell, and they are repopulate the urine, and the filamentous form which fail to grow intracellularly are not able to be cleared by the full nuclear cell, no matter what cell in the urine. But in the lining of the bladder, when the lining of the bladder is shed, it may be uptaken by the bladder cells, and then they form the QIRS, which is the quiescent or dormant intracellular reservoir. So these dormant QRs are very completely protected from antibiotic, and this may be one of the cause of the recurrent infection. So now the research is to find and to target IBCs and QRS so that you know uh, so that IBCs and QRS are not formed, and it's only is to will happen if we prevent the initial attachment of bacteria to the bladder wall. So this is an ongoing research on it. Thank you.